Hey everyone, my name is Aisha and I'll be presenting the modern breeding section of our group Cinchona tree presentation. The modern breeding of Cinchona focuses on improving the following aspects: queening yield, ensuring consistency, and supporting long-term conservation. Compared to other plants, the breeding of Cinchona has presented a lot of challenges. For instance, these trees are slow growing and can take years to reach maturity. They're known to be difficult to cultivate because of their environmental sensitivities. Medicinally valuable species generally cannot tolerate frost. They require cool climates with minimal temperature fluctuations and also dislike stagnant moisture and direct sunlight. As a result, Cinchona cannot thrive on flat land. The plant does best in rich, well-drained mountain soils. These preferences make traditional selection methods time-consuming and very inefficient. In early cultivation attempts, such as those in Madras, seedlings had to be shaded to protect them from sun exposure and retain ideal microclimates. Light pandals or fern canopies were used to do this. In addition to these environmental sensitivities, Cinchona also has a high tendency to hybridize, especially in its most potent species, making genetic stability a breeding challenge. Another complication in Cinchona breeding is the wide variation in quinine content, not only between species and plantations, but even across different parts of the same tree. Many of the early exported South American varieties contained as little as 1.5% quinine, making commercial cultivation economically unsustainable. However, in 1865, this changed when Charles Ledger smuggled seeds from South America. These seeds contained exceptionally high quinine content, between 8 to 13.5%. This breakthrough variety became known as Cinchona calisaya ledgeriana, and its widespread export eventually ended South America's monopoly on quinine. Historically, the approach to improving Cinchona strains involved selective breeding, planting seeds or cuttings from trees that produce higher level of quinine. This method was widely used during the colonial period, especially in places like Java, where Cinchona plantations flourished under Dutch control. However, the downside of selective breeding is that it relies heavily on observable traits, and quinine levels can only be accurately measured once the tree has completely matured and been harvested. Now that we've discussed traditional methods of breeding the Cinchona plant, let's move on to modern breeding techniques, which offer more precise and sustainable ways to improve cultivation. One key method is clonal propagation, particularly through tissue culture. This technique allows scientists to produce genetically identical copies of high-yielding trees under sterile, controlled conditions, ensuring consistent quinine content across plants and accelerating the production of new trees. Micropropagation, the technique used in clonal propagation, is a type of plant tissue culture that enables the rapid multiplication of plants using small tissue samples from a parent plant. These samples, called explants, are placed in a nutrient-rich medium under sterile conditions to promote growth. As the explants develop into plantlets, they are carefully transferred to new environments for acclimatization before being planted in soil. This method is especially valuable for producing large numbers of uniform, high-quality plants in a relatively short time. It also helps preserve desirable traits, such as high quinine content, in each new generation of Cinchona trees, making micropropagation an essential tool in modern breeding and conservation efforts. Another advancement is the use of molecular markers and genomic tools to support selective breeding. Molecular markers like SNPs are extensively used to identify genomic regions associated with key traits, thereby accelerating breeding process. Genomic selection enables the prediction of breeding outcomes by analyzing the genetic potential of plants, leading to improved crop yield, disease resistance, and stress tolerance. Furthermore, genome editing technologies allow for precise gene modifications, facilitating the development of new plant varieties with desirable traits. These advancements collectively contribute to the efficient and sustainable cultivation of plants like the Cinchona tree, ensuring a consistent supply of high-quality plant material for quinine production. By targeting these genes, Breeders may eventually be able to select or even modify plants for optimal production without having to wait years for the tree to mature. This represents a significant shift towards a more data-driven precision breeding. In addition to genetic breeding, modern conservation efforts are also being integrated into cinchona cultivation. Due to the historical overharvesting of wild cinchona populations, particularly in the Andes, these growing interests in ex situ conservation, maintaining genetic diversity in botanical gardens and research centers, and in reforestation projects that include improved varieties. These programs often incorporate community-based approaches, encouraging local participation in both conservation and cultivation, especially in regions where traditional use of cinchona is still practiced. While quinine may no longer be the frontline treatment for malaria, the cinchona tree remains a powerful symbol of plant-based medicine and the global exchange of botanical knowledge. Its breeding story reflects not just advancements in agriculture, but also the importance of balancing science with sustainability and ethical cultivation. Thanks for listening.